as our private sector panelists, let's say. And we have Sophie Aguerre from Rumigen project, Carl Kroger from Geronimo project, and Luca Fontanesi from Livestock project as a cluster member. And Luca will moderate the discussion, which we hope to have a fruitful one. So, Luca, I'm giving the floor to you. Please.
Okay, thank you. Thank you very much for inviting me to this event, a very important event in this framework. I start to share my screen. Just a second, and should be there. All right. So the main topic of my presentation will be focus on innovative strategies for sustainable breeding with a specific application and examples derived from molecular phenomics investigated in pigs. First of all, I would like to thank EULIFE, that is the cost action that I'm chairing. EULIFE is a European network on livestock phenomics, and also Livestock. I'm working into the Livestock project. So some of the results that I will present will derive from the activities that we have been carrying out in these two projects. Just a few words about where I come from. University of Bologna. Bologna is in Italy, in the north of Italy, and is the oldest university in the world, actually in the Western countries, to be precise. It was founded in the year 1088, and you are invited to come to Bologna and see how beautiful is the town. I come from the Department of Agriculture and Food Sciences, which has 10 divisions, and I work in the Animal Science division. I'm leading the Animal and Food Genomics group, a small group with very smart people here, and I cannot list all people here. They did a great job in producing the result that I'm going to present. So to start with, I want just to start with some basic concept coming from the European Network on Livestock Phenomics to introduce what we are doing in the Livestock project. EULIFE is a cost action. A cost action is not really a project, it's a networking tool. It has been funded from 2023 till 2027. And at present, the network includes 64 managing committee members from 36 cost countries, a total of more than 400 working group members from more than 50 countries. I'm chairing the cost action, and the co-chair is Thomas Norton from QLeuven. The cost action is divided in 4 working groups, and here just present the people and the working groups. Working group 1 is phenotyping technologies. That is one of the core of my presentation. Working group 2 is genome to phenome integration. Working group 3 works in computational resources and methodologies for data analysis. Actually, phenomics is a big data science. Working group 4 is very relevant. Economic impact, regulation policy, and society focus on animal phenomics. And the working group 5 is focused on stakeholder engagement, communication, and dissemination. This network of people led by many of people that you probably know. There is also Gina Katrick from FAB is involved, and there are also other group of people that are carrying on a very important part of the job in managing this networking tool. You can, of course, join the cost action if you are interested in livestock phenomics, and this is just a QR code, and of course you can Google just using the code of the cost action if you want to get more information, and you can follow the cost action through the social media. Well, we are developing this mantra, let's say, this way to say phenomics as a central dogma in animal science. Why? Because it is the central pivot, probably, around which most of the activities in livestock are then conducted in some way. Phenomics is a phenotyping part of the activities, and phenotyping is increasingly be recognized as a limiting factor in all application of livestock genetics and genomics. It is obviously clear. And but on the other hand, phenotyping is fundamental to routine and daily management of livestock population in order to optimize day-by-day activities in livestock production sectors, reproduction strategy, disease control, welfare of the animal, and so on. So these two parts are in some way unified with phenotyping. We need actually new phenotypes, new way to measure phenotypes, and new way to use the new phenotypes. This is clear. Phenomics, going back to the terminology, is emerging as a major new technical discipline in biology. Phenomics can be defined as a sort of ensemble of methodologies and technologies for the acquisition, analysis, and exploitation of high-dimensional phenotypic data on an organism-wide scale. The cost action that I'm sharing as a challenge, it is important definition when we start to run a cost action. We are requested to define an action challenge, and each word has a specific meaning. Even if this sentence is quite long, it is important to know that the action challenge wants to just produce novel scientific knowledge and application in the livestock production sector, focus on animal phenomena. Phenotypes can be classified in different ways, of course. There are different methods to classify phenotypes. There are ontologies and so on. We can divide phenotypes here into two main categories: external and final phenotypes, that are called performance, morphological, disease, resistance, and behavioral traits that are directly economic relevance in some way. And internal or molecular phenotypes that are the level or the presence or absence of different types of biomoleculars and their modification in animal biofluids or tissue and so on. So there are two main categories here. Here we will discuss molecular phenotypes. Actually, final phenotypes are determined by the contribution and interplay of many molecular phenotypes with multi-level relationships and their interaction with environmental factors. So this is the classical scheme that I usually use when I present this concept. There are many molecular phenotypes that are between the genome level, where genetic perturbation derives from the variability that are present in animal populations, and production traits. So in the middle, we have molecular phenotypes. There, of course, there are also environmental perturbation that in some way modify the different interactions. So going back to a real livestock, project, I'm involved in work packet 3, that is rebreeding livestock for resilience, and Birgit and I are co-leading this work package in real livestock, in the real livestock project. In the real livestock project, I will is focused on both cattle and pigs. I will talk about pigs only, and I will focus on novel phenotypes and genetics of adaptation to climate change. That is one of the tasks within Work Packet 3. In this task, there is the involvement of the University of Bologna, CSEC, Poznan University of Life Science, ANAS, the Italian Pig Breeder Association, and PIC. Going back to the general scheme between external molecular phenotypes and different levels, I will focus on the metabolite level. That is just one of the molecular phenotypes. Probably one of the yet unexplored level of molecular phenotypes where there could be a lot of additional progress. In real livestock, we have designed 2 main experimental schemes based on pig populations involved in large cohort designs that are used to link metabolomics to genomics and to heat tolerance. And another experimental design based on small cohorts used to link metabolomics to circadian rhythms, and might explain some adaptation to heat stress. Why this experimental design? Because we need to take advantage from let's say not really an advantage, but we need to value the amount of money that we have available for the project. So money is, of course, a limiting factor. So we have to design to merge 2 designs in order to take advantage from what could be possible to get from 2 perspectives in some way. So the expected outcomes from the 2 main experimental designs, the large and the small cohort, is this one. So this is the total number of metabolites that we are analyzing in the 2 types of experimental designs, large and small cohort experiments, 1,000 of metabolites. And the red circle includes a metabolite for which genetic variability and on the level in a biofluid, in this case is plasma, it means is heritable. And we can consider them interesting phenotypes for animal breeding, of course. And in the green circle, there are metabolites that might be involved in the response to heat stress, a small cohort. Some of them might not be irritable, other might be irritable. So the merge between the 2 experimental design could give this part of metabolite that would be very interesting in explaining the response to heat stress. I will just explain part of the information that we have obtained. The large cohort experimental design, we used targeted and targeted metabolomic platforms. More than 1,000 metabolites have been analyzed. Actually, we didn't just explore the single metabolites, but also the ratio between the metabolites. So we increased by 2 orders of magnitude the number of phenotypes. So more than let's say 200,000 phenotypes in some way. The population that we used were based on Italian large white pigs, Italian Duroc pigs from the Italian Pig Breeder Association, and Duroc pigs from PIC. So we analyzed these animals with metabolites, but of course we also obtained genomic information from all animals. We just published a few papers on that introduced that try to explain the informativeness of metabolites in the different cohorts that we analyzed. Just two papers as examples that I included here. The first paper, we wanted to identify if there could be differences between two breeds. Actually, this is the very basic element that say if there could be some difference in the metabolite profiles between breeds, it means breeds are genetically different, of course. So the difference could in some way the metabolomic profiles that explain difference between breeds that could be partially genetically explained. We used an on-target platforms here, and we identified 100 metabolites that could clearly separate the two breeds derived from here, represented by different dots of different colors. So in some way, the platforms works, and then we use this platform for a larger analysis and a larger exploitation of the information. In the second paper that we just published in Genetic Selection Evolution, we used the Italian large white and Italian Duroc pig breeds, and this is representation of the level of heritability of the metabolites that we analyzed in this paper, and this is the correlation between the heritability estimated using genomic information and pedigree information in the two breeds. So there is a high level of correlation between the two methods, and there is a broad variability of the level of heritability coming from just almost 1 to 0, of course. Here also there is another very interesting example that could be interesting to go into the details of a molecular phenotypes. There is a sort of relationship between the heritability and the number of carbon atoms or double bonds within different families of metabolites. Actually, we analyzed many metabolites that are different in terms of number of carbon atoms or number of double bonds within a family. So we are able to investigate also this relationship, and it's a very interesting relationship. Then we, of course, merged the metabolomic and genomic information, a classical GWAS, and here are the results, combined results, joint Manhattan plots that summarize the results of the GWAS in using single metabolites or ratio of the metabolites here. For the large white and the Duroc or meta-analysis combining the two population. And there are many interesting pigs, and actually just using a small number of animals, it is possible to identify SNPs that explain a large fraction of the genetic variability for the content in the plasma of some specific metabolites. Most of them are also nutrients, opening a very interesting perspective for nutrigenetic approaches. Here is just another way to dissect the information that we can obtain. Just an example. Here is just identifying the leading SNP in terms of associations, and putting in some way the so-called reverse GWAS that I call in this way, even it is not properly the reverse GWAS, but I just plotted the metabolites of different families for and the p-value for the top SNP for one of them. So there are interesting relationships that describe how in some way there is relationship between different families of metabolites, and within families different type of metabolites that are different in terms of number of carbon atoms and double bonds, describing very important pathways. Here is a two-dimensional profile based on metabolites ratio that can in some way further describe these molecular phenotypes and can give an overview of if there could be some difference between a breach or not in terms of effects into some metabolites. Again, here is so another way to represent this information. It is possible to use only metabolomic information to reconstruct pathway metabolic pathway into the plasma, and Gaussian graphical models can obtain high level of correlation between metabolites that are closely related to each other. That means that are very closely related in terms of metabolomic pathway. So there are just different in terms of one steps of metabolomic pathway. And so there is a possibility to correlate the effect of the QTLs that are being identified. So we are here. So in real livestock, we have identified many novel phenotypes that might be related to adaptation to climate change. Of course, I cannot go into all details of how we can interpret the information. And we are applying the information to develop genetic valuation models for pigs in order to include metabolomic information and, of course, the genetic correlation between these novel phenotypes and the external phenotypes. And we are also developing new breeding strategy for mitigation and adaptation to climate change. Of course, adaptation for the pig, mitigation is mainly for the cattle part. So metabolomics plus genomics may dissect biological mechanism, and it is possible to merge metabolomics and genomics for applications, and metabolomics and genomics can generate novel hypotheses. We are just at the beginning, actually. This is just the fraction of the metabolite that we analyzed. There is much more, of course, that is undisclosed, and there is a large room for molecular phenomics in this context. I take also the opportunity to mention that within the cost action, we are organizing the EU-LIFE stakeholder meeting, where phenomics will be the core of the discussion between many actors, many stakeholders, divided in different topics: phenotypic innovation, opportunities and trade, collaboration in action, and so on. The stakeholder event will be organized on the 11th of July in Leuven, when we will have the annual meeting of the cost action. Of course, you can register, and please, if you need additional information, come to me or also Gina or Thomas Norton. Again, phenomics is the central dogma in animal science, can join cost action And I would like to thank all the people of my group that did a great job in producing the data and helping me in all the stuff. And also Real Livestock and ULife, and also you for listening. Thank you. I think I can, yeah.
Yes, please go on, Luca.
No, okay, that's fine.
Thank you, by the way, very interesting presentation.
Okay, thank you very much. I think I need to give the floor to Karl Kroger from INRAE for his presentation.
Thank you,
So,
Luca.
Karl, yeah, the floor is yours. Thank you.
I will share my presentation. Can everyone see it?
Yes.
Okay, perfect. Thank you everyone for inviting me here and letting me speak today. I'm a second-year PhD student at Agroperitech at the University of Paris-Saclay, and my thesis falls underneath the Geronimo project and is in conjunction with INRAE and CSIF. Today I'll be discussing local breeds and landscape genomics, a powerful combination for detecting adaptive genes. Okay, so today I will be discussing specifically chickens, but theoretically what we're looking at can be applied to other species as well. Here is a map of the earliest confidentially assigned chicken remains across Eurasia, Africa, and Oceania. So in this project, they tried to identify the time frame in which the chicken arrived to Europe based on archaeological evidence. And they cited that the earliest directly dated chickens in Southern Europe were excavated in Italy from 2 Greek island colonies dating back to 776 to 540 BCE. So the chicken has been here in Europe for a very long time. And anything that's been around for a long time has had time to fracture and break into smaller subunits. And those smaller populations would be what we refer to as local breeds. Local breeds are a specific breed, a specific phenotype that has been shaped by the culture and history of a very specific region. So we can see the remains of local breeds within art, within historical legends and lore, and even modern today, some of these towns and villages that develop these local breeds and are maintaining these local breeds are still celebrating them with local festivals. But regardless of their historical and cultural significance, these local breeds contain a diverse genetic resources. So within chickens specifically, you can see a large variation in feather coverages, comb coverage, feather types, where the feathers are at, whether we have naked neck or fully feathered heads, and egg colors. And the differences between our local breeds phenotypically go on and on and on. A lot of these phenotypes were driven and selected by humans. However, knowing that these breeds were developed in a very specific region and maintained in a very specific region under a very specific climate, we have to ask the question, is their genetics also shaped by the climate? So this gets to the objective of my first chapter of my thesis. The aim of this study is to identify the underlying genes contributing to adaptation of European local breeds to the environmental conditions of their origin, using landscape genomic techniques. Landscape genomics is a field commonly associated with ecology and more specifically plants, but it's a rapidly advancing research field that's combining population genomics, landscape ecology, and spatial analytical techniques. So we're going to try to take our genome and see how much of that genomic variation we can explain by the environment while conditioning for population structure or what makes our populations different. What makes this kind of a little bit more difficult than traditional landscape genomics studies is that we have a huge influence of humans. So how are we separating our climatic influence from the human influence? Because we know that humans artificially manipulate the climate. We are building houses for the birds. We are protecting them from wildlife. We're providing them feed. We know there's breed isolation. So once we have a breed with a defined phenotype, farmers are reluctant to share and crossbreed. And then we also know humans are selecting for phenotypic traits such as growth, egg production, and then those visually appealing traits. So to try to account for this human influence, we go about it in 2 different ways. When it comes to artificial climate or what the birds are really experiencing. We try to account for this in our study design. And then when we're talking about breed isolation and human-driven selection, we try to include population structure within our statistical model. So first, let's kind of talk about what criteria we consider when building this study. So first, we want to make sure that the local breeds that we're choosing remain in the region of origin. We want to make sure that the breeds are experiencing the same climate that they are experiencing when they were first developed. They have a phenotypic standard for their breed. With minimal breeding on performance traits, we don't want to use any breeds that have been over-selected for any large growth or eggs. Minimal selection is key. And then we want to make sure that these birds are exposed to free-range housing, making sure they're exposed to the climate. Now, these are all very important when choosing what breeds to have, but we also need to consider the breed history. So how large are these breed populations? Are they very inbred? How old is the breed? And has the breed been recreated in recent history? So this is really important, especially with a lot of our local chicken breeds, because right around World War II, we see a lot of these local chicken breeds go completely extinct or very near extinction. And since then, they have been recovered, but a lot of that recovery has been through crossbreeding. So we want to try to keep breeds who have gone through as minimal crossbreeding and reconstruction as possible. And then finally, we just need to capture as diverse range of climates as physically possible. So for the project I'll be discussing today, we chose 26 breeds covering 6 different countries with a total of 1,334 individuals, which you can see on the map to my right. We have Iceland, Norway, Russia, most of our populations falling within France. We have Egypt and then the Ivory Coast as well. And we're averaging about 51 individuals per breed. Now comes the much trickier part, trying to include population structure within the model, trying to account for this breed isolation, as well as our human-driven preferences. To do this, we try to take into account population structure within the model. So we go ahead and run an admixture to break our genome into 2 different clusters. And using the results of this admixture, we then condition that in our model. Why did we choose a clustering of 2? So in this graph here, on our X, we see our different clusterings from 1 all the way up to 30, and the variance explained on our Y. And we see a huge drop in the amount of variance explained in our clustering 2. So this tells us that with 2 clusters, we capture a lot of that genetic diversity that's happening within our breeds. But not only that, we know from from previous papers and previous research that at this K of 2, we're capturing the bird's introduction into Europe. So to my right here, this is a paper published on similar data, and we are coloring these breeds based on their introduction to Europe. So these red dots are for Asian introduction versus the blue dots, which are considered more Mediterranean or European of origin. And so we know at a K of 2 that we're capturing at least the history up to when they arrived into Europe. Now, it's very possible and very likely that a higher clustering value is more beneficial, and that's something we're currently looking into to try and figure out how best to capture that population structure without over accounting for it. But this provides us with our final statistical model of 24,820 SNPs across our environmental variables, mean diurnal range, isothermality, mean temperature of wettest quarter, and elevation, conditioned for our admixture cluster. I will say that we pulled environmental data from WorldClimate, and we pulled it based on each breed's city of origin. And WorldClimate gives us multiple variables. I think it's 19. And we went ahead and did a variable selection to kind of simplify the overall model and choose the top 4 variables that explain the most of the genetic variation within our project. When it comes to the G by E, or genotype by environment analysis, we ran a redundancy analysis, or RDA, within the vegan package. The redundancy analysis is a multivariate statistical technique used to explore the relationship between two sets of variables. It's easy to think about this very similar to a PCA, and it's allowing us to take into account how the environmental variables are interacting with each other and not looking at each individual variable alone. And then our latent factor mix model, this was run in the LFMM package. And this is a regression model in which the response variable is a genomic matrix and the explanatory variable are our environmental measures. And this is very similar to a GWAS. The only major difference is that in the latent factor mix model, we are accounting for population structure. So as I go on to the results, our RDA analysis found that our 4 environmental variables accounted for about 4% of that overall genetic variation. And as I said before, you can kind of think of the RDA very similar to a principal component analysis or PCA. So we have our different RDA axes that are calculated as composing a fraction of each of our original climatic variables. So if I were to look at the first RDA axis, it's largely comprised of mean temperature wettest quarter, elevation, and isothermality. And it explains about 39% of that total 4% variation within the genome. And if I were to try to explain what all of this RDA axis is capturing, I would say that RDA1 captures seasonal temperatures, warm and wet. So its rainy season is when it's warm, so in the summer, at high elevations. And then RDA2 is mostly contributed to by mean diurnal range. So it's a very stable daily temperatures. I like to think of these axes as going from individual climatic variables to more environments. So this is allowing us to test for environments more so than just looking at the individual variables alone. We're also able to graph the RDA results. So here in this graph on the x-axis is our first RDA axis, so RDA1, and on our y-axis is RDA2. Each of these clusterings is a breed, and then I've colored these breeds based on the climatic zone to my right. The non-French breeds are in black. But what we see here is what we were hoping to see. We are able to remove some of that population structure based on feather color and phenotypic things like that, and more of that climatic influence. And we see our oceanic breeds clustering, we see our semi-oceanic breeds clustering, and then both oceanic and semi-oceanic together, versus our continental and our mountainous breeds. So they're graphing very similar to the effect that climates have on the genomes. And it's also important to know that our non-French breeds are following the same trend. So if we remember, our RDA2 is mean diurnal range, which a high correlation with mean diurnal range was associated with stable day and night temperature, so relating to Norway, versus Egypt in the desert, where you have big extremes between day and night. So they are falling roughly into where we're expecting them climatically. As we move on to our significant SNP identification, our 2 tests, our RDA found a total of 405 individual SNPs, total unique individual SNPs. It's important to note that we are testing linearly across each of these new RDA axes. And because of that, you can find the same SNP correlating with multiple axes. And then for our latent factor mix model, we found a total of 49 significant SNPs. And once again, you can find, you're able to find the same SNP across associating with the same climatic variable. When we compare the results from these 2 tests, we find a total of 8 SNPs that are overlapping. This isn't a ton, but it is very exciting to find. And we went ahead and investigated these 8 overlapping SNPs just a little bit more. So the first one I will highlight is a SNP that fell within the gene ZNF 804B, and it was associated, or we found it associated with elevation. So on the map here, you can see that across the landscape, You have a map of the elevation. So the Alps here are highlighted in red versus valleys and coastal areas in the dark blue. Each of these dots represents one of our breeds. And then the number is correlated, or is the mean allele frequency for the SNP found within this gene. And it's not super clear, but there does seem to be a small trend or some sort of pattern starting to appear where our higher elevations have a higher minor allele frequency versus our low-lying or low elevation populations. But don't just take my word for it that this could be adaptive. Other research projects have also identified this gene to possibly be adaptive. So it was found to be between mainland and island sheep in Greece to possibly play some long-term local adaptive benefit. And then among greater teats, it was found to be a long-term adaptation to high altitude environments. So like our elevation. So this is really exciting because it does suggest that what we are finding could very well be adaptive. But you have to take everything kind of with a grain of salt. So with this, I'll go into another one of those genes, the BCO2, which was found across mean diurnal range. The BCO2 gene is very common and very popular within poultry. It's used and really associated with differentiating between yellow skin and white skin. It's something that we know already is under a lot of selection pressure, especially from humans. And we see that outside of France, the frequencies are very high, where within France, it's very low. What this means, we're not really sure yet. We're not sure why we're finding it, if we're finding it because of some human-driven selection, or if this is truly adaptive. There is some papers suggesting that there could be an adaptive advantage to the alleles within this gene to account for high demand for carotenoids, where you're in an environment where carotenoids are in low supply. But knowing that it's correlating between white skin and yellow skin, which is under preference and selection by humans, it's hard to tell and be really confident about its association with the mean diurnal range. However, what we can be confident about is that this is under selection. So regardless of why we're finding it, we are still able to find genes and SNPs that are under selection. And with that, I just want to bring everything back to our local breeds. So according to the FAO, of the local breeds that we know, which we know very few, about 80% of those are at risk for extinction. So I try to highlight here that within the genome, there's a lot of potential within our local breeds. There's a lot of secrets hidden there that we can try to leverage to help us get into the future and to start breeding and adapting our birds for our future climates. However, with them most all being at risk of extinction, once they're gone, they're gone. So this should help highlight that we need to really focus on maintaining and preserving our local breeds. And with that, I would like to say thank you to everyone. Thank you to the Geronimo Project and all my supervisors. Thank you.
Thank you very much, Carl. Very interesting presentation. And you have shown value of animal genetic resources here. And now I think I have to give the floor to Sophie Aguerre from IDELE. Sophie, the floor is yours. Thank you.
Thank you. Now, do you see my presentation?
Yes.
Thank you. I work for IDELE, so the French Livestock Institute, and I'm also part of the bovine genetic team in Jouy-en-Josas with Alliance and INRAE. Thank you for giving me today the opportunity to present this work. I will present today a study on the impact of heat stress on production, other health and reproduction in dairy cows in the Netherlands, Spain, and France. It was a work conducted in the HoloRuminant project. So we, as you can see, many partners worked with me on this project. Partners from INRAE and INRAE in Spain, from Wageningen in the Netherlands, and from INRAE and IDELE in France. So in the context of climate change, as you know, it will be characterized by a global rise of temperature and also by extreme events such as heat waves that will become more frequent, longer, and more intense. And this will affect dairy cattle because they are particularly sensitive to heat stress. It will have impacts on their physiology, health, welfare, and zootechnical performances. So in this context, the HoloRuminant project aims at providing breeding tools to achieve a better adaptation of cattle to climate change. And today I will present a work that was performed in the work package 3 of the project, which had for objective to measure the impact of heat stress on the performances, first of dairy cows, to evaluate the impact of heat stress on their performances and the genetic variability for tolerance to heat stress, and to evaluate the impact of heat stress on trade-offs between production and reproduction. And then we also studied the impact on their daughters, so to evaluate the impact during the gestation of heat stress on the spring phenotypes. So 3 different breeds were studied. Holstein in the 3 countries, and then Montbéliard breed in France, and the INRAE in the Netherlands. So we worked with large-scale national datasets of zootechnical performances in first and second lactation. So for production traits, we used taste day records on milk yield, fat yield, and protein yield. For other health, we focused on the somatic cell score, and for reproduction, on the conception rate at first artificial insemination. So you have here a table with the periods and the number of cows included in the study for the different countries. So you can see that we worked on recent years, so between 2010 and 2020. And the data sizes were a bit variable from one country to another. We also used pedigree and daily weather information that were provided by national meteo agencies, which were associated to each herd using their zip code or farm coordinates. And to measure heat stress, we use the temperature humidity index which rely on the average daily temperature and the average daily humidity. So it takes into account temperature and humidity during the day and during the night. In the first step, we studied the impact of heat stress on dairy cows' performances at the population level. So for this study, each lactation was analyzed separately. And we have a model, so one for production and somatic cell score, and one for reproduction. So the performances of the cow was explained by a THI effect, different fixed effect, and genetic effect. A genetic effect and a permanent environment effect here, because we have several records for these traits. And for reproduction, we have one performance per cow. So there is also a THI effect, and then the same type of model, but with only an additive genetic value. So for the THI effect, we took it as a fixed effect for this study in the model. And for production and somaticity score, it was a THI average within 3 days before test day. So the 2 days before test day and the test day. And for reproduction, it was average on 8 days after artificial insemination. So it's traced on the short term around the performances. So on this slide, you can see a graph representing milk yield depending on the value of THI, with in green the results obtained in France, in orange the results obtained in the Netherlands, and in black the results obtained in Spain. And you have here some information on what the value of THI represents in Celsius degrees. So what we found was a similar rate of decline with daily production loss of 6 to 11% between THI 50 and THI 70. So THI 50 is around 9 Celsius degrees on average, and it's more or less the current conditions at least in France and the Netherlands. And THI 70 will be around 20 to 25 Celsius degrees. So we had similar rate of decline, but different threshold. As you can see, in France, the threshold is quite lower than in the Netherlands and in Spain. So we have more differences among countries than among breeds. In France, for example, the threshold was quite similar between the two breeds, Holstein and Montbéliard. And the same was observed in the Netherlands for the two breeds. So our hypothesis to explain this different threshold was more in relation with the different farming conditions in the different countries. So which could explain different exposure to outdoor weather conditions. In France, there is also possible confusion between the THI effects and the transition periods from indoor to pasture ratios. So the THI effect would absorb a bit of these effects. And for Spain, there is an hypothesis around the mitigation since housing conditions are already a bit adapted for cows in Spain, and the possible acclimatization of the cows. Here you have a similar graphs, but for the consumption rate. So for these traits, we found thresholds that were more homogeneous across the different countries. But we found a steeper decline for French and Dutch Holstein cows than for the Montbéliard here and the Spanish cows. So again, the hypothesis around these different results are around the acclimatation and housing conditions for Spanish Holstein cows, and maybe different genetic abilities for the French Montbéliard cows. So in the second step, we had a look at the genotype environment interactions, so to look the individual response of the animals. So just a quick introduction to the methodology here. So the objective is to the objective is to have an idea of the evolution of the genetic value of the animals along the THI gradient. So to do that, we use random regression model. So the interest is that we can have the evolution of the genetic value along the environmental gradient, in our case, which was the THI gradient. And with random regression model, we can estimate the genetic values of the individuals along this gradient. Whereas in a classical model, we have a unique genetic value for each individual, which is constant whatever the environment. So for this part of the study, the model included fixed effect and then a genetic effect and a permanent environment effect. And you can see that here we take into account THI as the gradient to estimate our genetic values. The E-stress model, the E-stress effect was again 3 days before test day for production and somatic cell score, and 8 days after artificial insemination for reproduction. So you have here a representation of the evolution of genetic correlations for protein yield and conception rate between THI 50 and over THI values. So for production and somatic cell score, I put here only the example of protein yield, but it's the same for the different traits. And high correlations between THI 50 and over THI, indicating little to no genetic environment interactions. Whereas for conception rates, we have moderate genetic correlations after a breakpoint here in the Holstein populations. And you can see, for example, here for Spain, or for Montbéliard here, genetic correlations that become quite low, around 0.5 and 0.4, indicating the existence of genotype-environment interactions. We also had a look at how E-stress will impact the rankings of the elite reproducers. So what we observed here is an evolution of genetic values along the THI the THI gradient. You have in blue the top 10 sires of Montbéliard in the Montbéliard breed, and in red the 10 the 10 sires that were at the bottom of the ranking. And you can see that for production, we have differences between sires that are reduced, whereas for conception rates, the differences are amplified. So it indicates that for production, the cows with the highest level of production will be the most impacted in the future by E-stress. And for conception rate, the cows that have the most difficulties to reproduce will have even more difficulties in the future. But what we also see in these graphs is that we have different slopes of decline between individuals. So these results indicate that some animals are more tolerant to E-stress than others. So it will help us to select some animals. The next part of the project focused on the evolution of trade-offs along the THI gradient. So actually, we already observe unfavorable genetic correlations between production and functional traits, so health or reproduction traits. And we may anticipate that these correlations will become even more unfavorable in the future warmer conditions. So we had two main objectives in this part. It was to predict the trade-off in future conditions and predict the effects of current selection on this trade. So if we first focus on the trade-off in future conditions, you have here the trajectories of the genetic correlations between protein yield and conception rate at the same THI. So in blue for the Holstein French population, in red for the Netherlands, for the Holstein in Netherlands, in green for the Holstein in Spain, and in purple for the Montbéliard in France. So as you can see, the results were not fully consistent across population. In Spain, we observe a trajectory with genetic correlations that become positive. So the hypothesis is a possible acclimatization of the animals, where the animals that are able to maintain high production levels are also the animals that will be better adapted to heat globally, and so might be showing higher fertility results. Whereas in France, the genetic correlations remain more or less stable under heat stress conditions. And in the Netherlands, the correlation seems to become more unfavorable along the THI gradient. So it could be either an effect of the model or an indication that selecting for good production and good fertility will become more difficult in future conditions. And then if we try to predict the effect of current selection, we had a look at the trajectory of genetic correlations between protein yield at THI 50, so actual conditions, and the conception rate along the THI. So here you can see that the results were more consistent between the different countries, and they seem to indicate that the current selection on production has a limited impact on future reproduction abilities, but still slightly negative.
Okay.
And again, the impact is stronger in the Netherlands than in France or in Spain. So from these results, the recommendation is to adapt the breeding objectives for the future with higher weights for functional traits. And then quickly, I will present the impact of in utero heat stress. So the objective here was to assess the impact of stress at different stages of gestation on milk production and conception rate in Holstein and Montbéliard cows. So in France, we found limited impacts of maternal heat stress on progeny traits, with an average of loss of production of 1% of the average milk production. And in Spain, the evidence of an impact were clearer, with third generation cows showing noticeable declines in fat and protein yields, increasing with lactation number when their dams, grand dams, or great grand dams experience heat stress during the gestation. And the maternal environmental effect on cows' production traits was observed up to 8% of the genetic variation, with again an increase in impact according to the lactation number. And for both countries, the negative impacts of heat stress seem to be more during early gestation, in particular for protein and fat yields. So to conclude with the main results of this work, higher temperature will be associated with a decreased milk production, an increased risk of mastitis, and a reduced reproduction performances. Within traits, few genotype environment interactions were observed for production and somatic cell score, but they were stronger for reproduction. And we observed the variability between individuals indicating that some animals are more tolerant to stress than others. And it is important to adapt the breeding objective with higher weights on functional traits. And the impact of in utero stress was of variable magnitude, but the results all suggested a negative effect of stress in early gestation. So I would like to thank all the Rumigen partners, in particular those who worked with me on this work package, and thank you for your attention.
Okay, thank you very much, Sophie. Thank you, and I think now I give the floor to Larissa, I think, for additional
Yes, thank you, Luca.
elements.
Just
Thank you.
brief reflection on my side, representing the industry. I would like to thank all the speakers. Very interesting presentations and very relevant to the future of agriculture, the direction that we are going. I think that with Luca's presentation, we had a very nice example of new phenotypes and how as we evolve within this industry and within agriculture, we need to become very creative when it comes to phenotyping strategies, but also to what the role of the phenotype is. And I agree with you 100%, Luca. I think that phenomics is the central dogma. We're not going to get anywhere without phenotypes. So also as the industry, we need to become flexible and make use of the new technologies that we have available, but also learn exactly how they are going to play a role in the future for farmers in particular. I'm sure you all know it's not that easy to go to a farmer and say, this is what you need to do now. So we always need to keep that in mind. I think the whole sector, not only the industry, that we need to take the farmer into this journey with us. Otherwise, we're not going to get anywhere. Very interesting presentation from Carl as well. I think that the role of these local breeds is also something that we're going to have to rethink as we move towards the future. As the industry, we have been focused on a few particular breeds historically, but not only they offer a huge potential when it comes from a genetic diversity perspective, but also we, again, going back to the farmers, we need to be sensible about the value, the cultural value that these local breeds have in particular countries. But again, yeah, also a challenge for the industry. Always think we cannot have breeding programs for all the breeds. So it is something, of course, that we need to keep in mind and take this along our journey for the future. And well, I am a dairy geneticist, so of course, Sophie's presentation spoke a bit more closely to me. Very interesting work on the adaptation of heat stress for dairy cows. I think moving forward, as the focus has been so strong on Holstein breeds, but we also see a lot of challenges on the Holstein breed, a lot of challenges when it comes to heat resilience. But we also need, what I got from your presentation the most is that we need to take production systems into account as well, very strongly, as your results show clearly that the differences among breeds, between breeds was not as strong as the differences between countries. So that was very interesting. And again, as the industry, we need to be very sensible about solutions that are going to be applicable not only to the animal population, but also to each individual production systems. Otherwise, we can't get really far without the agreement of the farmers and being sensible about their production systems. Yeah, I think that's my reflection. I think we have some hard work in the agricultural business as we adapt to this new version of what the world is going to look like. It's an ever-changing world, but it's good to see so much good work being done. And I think we always need to be in touch. I think the industry and the academia need to work very closely because individually we're not going to get very far with the innovations. But thank you. It was really interesting.
Okay, very, very good. Thank you for your comments, for your considerations. Just another comment. I think the industry in animal science in general, I mean, not only animal breeding, should recognize that phenotyping is becoming a key issue. That is clear. And as animal breeders, we need to take advantage from what's going on phenotyping for other purposes. That is clear. So this could be a joint, let's say, initiatives to try to take advantage from the progress that PLF, for example, is developing. And we need, of course, as you mentioned, be very creative to obtain or to use, to extract, to elaborate, to analyze in different ways the phenotypes that could come from unexpected, sometimes applications. So this is could be quite challenging, of course, and could be different depending on the species, of course, on the production systems. So we probably need to talk to farmers again. It could be difficult sometimes. I fully understand. But this is something that could raise the attention on the animal science sector that is, let's say, in this period, not very considered from the public, I guess.
Absolutely. No, I fully agree with you. I think we are constantly on a position of defending ourselves and defending our industry. But yeah, that's a whole separate discussion. I do agree. We have to take advantage of technology and all these new phenotypes, but also understand how we can use them moving forward, right? Mm-hmm. And as you said, the farmer needs to be taken along with us in the journey. And it's usually it's about showing the value to the farmer, right? I think it's about showing the farmer that there is something for him. And farmers care about welfare. It's not only production. Farmers care about welfare. Farmers want their herds to be healthy and sustainable. If you talk to farmers, you see most of them are very innovative and have a very progressive thinking that is highly focused on the welfare of their animals.
Yeah, of course.
So we show there is value to whatever new technologies and whatever new phenotypes that we want to implement to them and to their herds. It's then it's a no-brainer. But the challenge is getting there, I think, is showing them the value.
Yeah. Thank you. Are there any other comments, questions from the audience, from the public? Chakra.
Luca, just coming to Larissa's first comment on that the research and the market or the commercial companies should align more about these research topics. In fact, I just wanted to highlight for our audience, if they're not aware of that, we have this farm animal breeding and reproductive European technology platform called FarmerTP, which is led by knowledge institutes and academia collaborating together with the private sector. So this is a kind of like public-private partnership to make a strategy for the coming years on what we should together focus for a more sustainable and agreeable, let's say, livestock sector, also taking into consideration not just research and market, but also the consumers on one hand and also the farmers on the other side. And I think it was quite a legitimate comment to mention farmers because most of the time, I mean, coming from a very farmer focused past, we always talk from a very high level with really undermining, in fact, what the farmers are doing every day there. I mean, they are really actually, especially like I also come from dairy sector, so I know that they wake up at 4 o'clock together with the sunrise or even before, and they work really hard and they're doing their best. But I think they're most of the time quite forgotten in many of the parts, also by the consumers. Like, I mean, if there were no farmers, we would be all going hungry. So we really, I think, should also have very good routes to communicate with farmers, engage them in these discussions. Unfortunately, it's not always easy, especially from a European level when you have the language barrier, but well, AI is coming there. Another question, in fact, maybe not so relevant about very specific genetics because I'm not a geneticist, but well, you probably all of you read that even like when you are talking with these like now artificial intelligence, even saying like thank you or please, it takes a lot of energy. It uses a lot of electricity and it is not sustainable on the long run. So with all these PLF technologies, phenomics, phenotyping technologies that we might be maybe switching into to have better accurate results. But also, I mean, while trying to make farms, livestock production more sustainable, do we, on the other hand, by using a lot of, let's say, electricity or other resources, natural resources, to get these data being processed, how do we balance it? Or do we have to also consider that now before having the problem to avoid it?
Yeah, well, probably I can try to start to answer your question. Yes, actually, we need a lot of research into this field. This is clear. I mean, because the potential is huge. But of course, there are some potential issues related to the use of big data that are not really considered sometimes. And of course, we need to do many research activities to find a sustainable way to use novel approaches, novel technologies, and innovation in this field. So we need research. This is clear in this context. And I think we need to push for having calls topics focus on that, because this could, I already mentioned, this is a central dogma in animal science. This is clear for me. And also I hope it is clear for many others in this context. So we need to distribute this concept, I think, because otherwise the risk is there will no research in this context. So this is something that we need. We need to explore the potential, but of course in a sustainable way. I think there are some questions on the chat. We can take a look at the questions. I read, how do you see this resilience indicator performing under tropical breeds and conditions where THI is persistently high and other stressors like parasites and limited feed are common? This is a question from Agnes Miamial. I think could be addressed mainly to Sophie, I guess.
Yes, it is difficult to answer. Well, in our case, the dataset at high THI was quite limited, so it is difficult to really answer to compare it to THI under tropical conditions. But I think there are also studies conducted in these conditions. They shows similar impacts on production and reproduction. You have, for example, works conducted in Florida, I think. They showed also an impact of heat stress on the production of the cows and also on the production of the progeny. So this work might be an beginning of an answer. But yeah, I guess that genotype environment interactions are maybe even stronger in these conditions.
Okay, thank you. So I think the the answer is complex, of course. We need to recognize this. And of course, research activities are trying to find solution also for this question. There is another question on the chat, is addressed to Carl. I would like to ask Carl if he could speak a little about the number of individuals that he had in his study with chicken, and what would be the challenges to replicate his study with small populations, especially population from conservation centers that usually are kept at the same environmental unity, one big farm center.
Yeah, I would be happy to talk about this question a little bit. And it's a really good question because a lot of our local breeds, it's really hard to get out to the farmers and collect the data from individual small farmers that are keeping these breeds alive. And oftentimes when we're getting samples on these local breeds, they're maintained by some conservation center. So I think to first talk about the number of individuals, I think it's really important that you pull a number of individuals that really captures the whole genetic diversity within that breed. If you have a really inbred breed, you probably don't need as many individuals versus a very diverse breed, you need more. You just want to make sure that the number of individuals you are using really is representative of the whole population and the whole breed as a whole, well, the breed as a whole. And then this concept of pulling breeds that are all coming from the same conservation center. This is a difficult question because in the modern time, they are all experiencing the same climate, but historically they would have been adapted or developed in various climates. And specifically for my project, we like to think that we are trying to find ancient adaptation or adaptation from like the development of the breed. So theoretically, if the breed has not gone through any new selection pressures or any new strong selection, we could still pick up on signatures of adaptation to climate related to their initial climatic zones. But what's important is that when you're running the study, you aren't using the current climate data, but the climate data from the region where they were first developed. But I can't 100% answer what you'll find because the question is, when is adaptation happening? Are we collecting adaptive signatures from the past when they developed, or is it more recent adaptation from what they're experiencing now? And that I'm not sure.
Okay, then thank you. Thank you very much for your comments. And are there any other questions from the audience? I wonder if there could be other questions. Or maybe I just have a general question that is related to animal genetic resources in general, not only chicken. How do you see the value of animal genetic resources in some way for taking some information useful for other application in more let's say cosmopolitan breeds? What do you think, Carl? And maybe also this question could be also addressed to Sophie, but also to Larissa. How the breeding industry see the value of animal genetic resources for taking some information useful for implementing let's say novel strategies in more cosmopolitan breeds. What do you think, Carl?
I think that's a good question. What do you mean by cosmopolitan breed?
Cosmopolitan breeds, small populations are not cosmopolitan breeds. Cosmopolitan breeds is Holstein, for example, or maybe commercial pig breeds or something like that. I mean,
right. So what I see with the results I'm getting right now is with these SNPs, with these genes I'm identifying, more to help guide the production, our production breeds, to be able to implement these genes to try to help our cosmetology breed our production breeds to be more efficient. Because I don't think it's really practical to expect a crossbreeding with our local breeds, which are very unproductive. It's a lack, like not possible. And there is a thought that we could do some sort of gene editing or gene splicing with our local breeds, but I don't think that's anywhere in our near future, especially with being accepted in the public. So in terms of the impact it has on our cosmetology breeds, I think it's more of just helping guide our breeding strategies, helping guide making decisions, because our cosmetology breeds aren't experiencing the real world. When we look at our Holstein, they don't have a chance to adapt because we're using artificial insemination. We're taking semen from all over the world. And so it's just really, they don't have the chance that some of our local breeds are having. So I think we can use the local breeds to really, I don't know, leverage the environments that they're living in to give us information on what to do with our cosmetology breeds.
Okay, thank you. Just a flash comment from Sophie and then final comment from Larissa.
Yes, I quite agree with Carl. In the Rumigen project, there are also studies on fields like epigenetics and how to manage genetic diversity, and also on gene editing, to try to imagine how we can implement the breeding tools in the future. But there is the question of the acceptability by the public and by the farmers, which is also studied in the Rumigen project. So I guess it will give us some keys on what we can do and cannot do.
Okay, thank you, thank you. Larissa, what do
Yeah,
you think
no,
about
I
that?
100% agree with Sophie on that one. We need to, I think they are a very important resource, and we need to find ways to preserve the genetic diversity that the local breeds, of course, bring. And as I mentioned in my reflection, also respect their cultural importance and regional importances. But there's plenty that we could learn from them. I think even the comment about in the chat, the question about the tropical breeds and this indication of being more resilient under harsh conditions. There's lots that we can learn from them, but we need to take into account what can be done when it comes from a society perspective and how to implement some of those benefits into our mainstream breeds. So it's interesting to hear that that there is this sort of reflection in the Rumigen project, because I think we do need to understand better what can be done that will be accepted.
Okay, thank you, thank you. There is a final question on the chat. Research has shown that resilience is very important in animal breeding. What do you think is the reason why breeding companies are not including it in their breeding goal until now? Maybe I could address this final question to you, Larissa, or what do you think about that?
I think that's probably I don't know if this question is specific for a country, because countries have different breeding goals. So I think generalizing it and saying that the animal breeding industry is not including resilience in its breeding goal is a bit of a stretch. In the Netherlands, we have a resilient breeding value, for example. So I think that many countries are very aware that this is a very important trait. If you look at the Scandinavian countries as well, their breeding goals are very balanced. So I'm not sure exactly what this question is addressing. Sorry, if you could clarify, then yeah, but from my perspective, there are plenty of countries that have resilience included in breeding goals.
Yeah.
Just add something real quick. We're looking at this from the fact that we're breeding currently, but our breeding has been going on for decades and decades, and research is constantly evolving. So it's not that we didn't start selecting for resilience. It's just we didn't know we should be looking at it. Like, I think as we're continuing to add more and more things we're breeding towards. And so it was just, we got to a point that we realized, wow, you need to really focus on this. And that's where we're at now.
Shagla?
Yes, maybe I can address Tomasi to our Code FAB, our responsible breeding website. Remar can share the link because we have been running a good code of good practice by FAB. FAB is the organization that has been representing the breeding companies in Europe, animal breeding companies in Europe for decades now. So we already have a code of good conduct, which you can check from the link Remark put. And you can see that we are, I mean, how the shift is in the breeding goals since, I don't know, 90s until now, or even past in the last 20, even 20 years. So there is a focus on animal health and welfare, a big focus. And in all the species, we have this focus. Please just have a look at it. And I'm sure if you have questions, just contact us. And most of our members are committed to these responsible breeding goals.
Yeah.
Thanks.
So, no, thank you very much for this final remark. The breeding industry is changing. Actually, has already changed the strategy since I think several years. So it is clear that there is in all breeding schemes that are in advanced structures, there are resilience elements that are considered carefully. Okay, I think we need probably to close. So,
Yes.
Chagla.
Thank you very much, Luca, for moderating the session. And thank you all, Sophie, Carl, and Larissa, for joining us with your comments and presentations. It was really, I think, quite fruitful, and we had a lot of interest. And thanks all our participants for joining us. We will be having a third episode of EcoGen webinar series in May. So just please keep an eye on our website or on social medias. And have a nice day.
Thank you.
Thank
Thank you very
everyone.
much. Thank you.
Bye.
Bye.
Bye.